Hello, sir. Myself Sri Lakshmi from the Department of Biotechnology, Section 2 BTA. Today, I am going to enact a role play of lipase. Hi, I am lipase are a family of enzymes that catalyzes the hydrolysis of fats. I am also known as the triacyl glycerol ester belongs to the subclass of esterases. I am the enzyme which catalyzes the hydrolysis of triglycerides to glycerol and free fatty acids over an oil water interference, and I am present in the body because I'm useful to break down the fats in food. So I can be absorbed into the intestine, but that is my initial digestion phase occurs in the small intestine. Also, I plays a very crucial role in digestion, transport of processing of dietary lipids in most of the organs. I was found by the scientist of Claude Barnett in the year of 1848. He would eventually attribute these reactions to an enzyme that was later named as lipase in the year of 1856, which is commonly found in the pancreatic juice. Animal pancreatic juice extract were traditionally used as the source of lipase for commercial applications. Source of lipase is generally in the stomach and pancreatic juice. In plants, source are present in the castor bean and oats. The lipolytic activity of the oats is 5.6. So the castor beans are difficult to obtain, so maybe an impractical commercial source of lipase. However, the oats are abundant. I am an enzyme which is present in the body, and I am used to break down the fats in food so that they can be absorbed easily into the intestine. And I am a may also found in the pancreas, mouth, stomach. AMP catalyzes the hydrolysis reaction and break down the ester bonds of lipids, fats, and convert into fatty acids, glycerol, and alcohols. Lipase is a group of fat splitting molecules which is commonly found in the blood, gastric juices, pancreatic secretions, intestinal juices, and also found in some of the adipose tissues. Enzyme who is involved in various biological processes which range from protein metabolism of dietary triglycerides to the cell signaling and inflammations. I am an enzyme who is confined to very specific compartments within the cell while the other work in extracellular spaces. Some of the locations of where I release and corresponding to the functions are given below. They I mostly secreted in the pancreas, and because the conversion of triglycerides in the presence of mine converts into the monoglycerals and fatty acids. Fungi, bacteria are also secrete the lipases, which helps in facilitating the nutrient absorption from external source. And whereas in some cases, the pathogenic microbes are also secrete the lipases for invasion of a new host. Whereas in cell biology, I play a very important and crucial role within the phospholipids that comprise the most of the biological different type of lipases, which are nothing but hepatic lipase, lipoprotein lipase, hormone sensitive lipase, and gastric lipase. The hepatic lipase and the lipoprotein lipase are situated in the location of endothelium, whereas the hormone sensitive lipase is located in the intracellular, and the gastric lipase is located in the digestive juice. The main difference between the hepatic lipase and the pancreatic lipase is hepatic lipase is mainly expressed or situated in the liver cells known as hepatocytes, whereas the gastric lipase is formerly secreted by the gastric cells that are found in the mucous lining of the stomach. And the gastric lipase is mainly responsible for the digestion of extra lipids, and most of the lipase function is determined by their structure, as many of the common lipases are conventional lipases. That means they share a common protein structure and catalytic mechanism of the lipases. Classically, lipase catalyzes the hydrolysis of triglycerides. That is, one molecule of triglycerides plus water gives rise to the fatty acid and glycerol. As we know that lipases are serine hydrolyses. That is, they function by transesterification generating an acylserine intermediate. In most of the cases, I found at the position of the glycerol backbone in a lipid substrate. A diverse array of genetically distinct lipase enzymes are found in nature, and they represent several type of protein folds and catalytic mechanisms. However, most Most of the enzymes are built on alpha beta hydrolase fold, employ a chirotrypsin like hydrolysis mechanism, usually done by a catalytic trade, which consists of a serine nucleophile and a histidine base and an acid residue, usually the aspartic acid. The mechanism is done as shown in the figure. The schematic diagram represents that how I plays a very crucial and essential role in every part of the human body. That is, in the stomach, due to the gastric juice produced in the stomach, the lipid digestion occurs. Whereas in the liver, here I store it in the gallbladder and further release it into the small intestine to help the digestion and absorption of lipids. And I am also found in the small intestine because to perform the function of digesting lipids very efficiently, and later the product is excreted as the fetus. And as I already mentioned in the pancreas, I release it into the small intestine to break down the fats of triglycerides into the monoglycerides and fatty acids. I am an enzyme who mostly found in your pancreas and helps to digest the body fats. However, the high level or the low level of lipases may you have a problem with your pancreas, that is the acute pancreatitis. So the acute pancreatitis conditions can be seen here in the picture. Thus, to maintain the moderate level of lipases in the body, there are some few things which are the uh, which helps to the secretion of lipases in your body are papaya, honey, mango, banana, avocado. This picture shows the various daily life applications of mine. In industrial uses, I can be found in the milk industries because since ancient times in human practices, I'll be very helpful in the uses of cheese fermentation as well as in the yogurt. And I plays a very crucial role as a biological catalyst for baking, laundry detergents and other products that deals with the household applications. And also, lipases are environmentally friendly and are safe to use. So I plays a very important role in the bioreactors also. And as the diagram shows, I may also found in the pharmaceutical industries, medical purpose industries, chemical, food and the textile industries, paper, leather, as well as the in the water treatment also I plays a very important role. Thus, I'm not only useful in the biological applications but are also useful in the various industrial applications as well. Thank you.